There's preclinical data and publications that show that the use of ibrutinib may improve the function of T cells, and most of that data is in CLL. But we thought that by giving ibrutinib prior to the collection of apheresis for the manufacture of Tisagenlecleucel or tisa-cel, that we could improve those T cells in these diffuse large B-cell lymphoma patients in the same way that that sort of preclinical data is showing, or observational data. And so this clinical trial gave ibrutinib in two ways for patients with relapsed refractory diffuse large B-cell that were going to get Tisagenlecleucel. One schedule was to give the ibrutinib weeks ahead of time before apheresis collection. The other schedule was to give it after collection. And so, we're able to sort of assess whether the ibrutinib is affecting the CAR T cell function after infusion, or if it's improving the T cells that we get from the patients. And the results are very interesting. Essentially, what we found is that when you give a ibrutinib ahead of time before you collect the autologous CAR T cells, we were finding that the T cell products were more sort of central memory-like and more naive or stem cell central memory-like, perhaps because of the ibrutinib that was administered. We also found there were more CAR T cells manufactured in those patients that got the ibrutinib lead-in prior to collection of their T cells. And the viability of the CAR T cell product was also improved. So there's very small numbers in this study, but I think it's encouraging that at least for patients who could wait and get sort of a lead-in of ibrutinib, it may help correct some of the dysfunction of their T cells, either due to prior chemotherapy treatments or because of T cell dysfunction driven by the diffuse large B-cell lymphoma itself.